on this episode of the WildFed Podcast.
Viruses do exist. They are the most plentiful entities on the planet. If you took all those viruses and put them end to end, they would go 100 million light years into space.
What? Wow.
If you could take all the viruses off the planet somehow, life would probably end in a few months. The host took this virus protein and used it to build the placenta. So if it weren't for that virus, we'd be laying eggs.
Episode 28 of the WildFed Podcast, The Wonderful Elegance of Viruses with Vincent Racaniello, is brought to you by Surthrival. Surthrival's product line was designed and developed for moments like these. Our concept was always thrive through any circumstances. To that end, we've created a line of supplements that empowers your immune system. Remember, these nationwide shutdowns were never designed to keep you from getting the virus forever. Rather, they were just meant to delay your exposure, the so-called flattening of the curve. At some point soon, these stay-at-home orders will end, and many of us will have our immune system challenged in new ways. Make sure you leave home with a stronger immune system than you started with. Surthrival's colostrum, medicinal mushroom formulas, and vitamin D3, K2 are all designed to support a robust, adaptable immune system. Find scientifically validated nutrients and superfoods that empower your immunity at surthrival.com. And I want to give a big thank you to everyone who's gone to our YouTube channel or website and watched episode 1 of the WildFed TV show. And to all of you who've purchased season 1 or the season 1 Director's Cut bundle package, I want you to know how much we appreciate you. Your support is making this show possible. If you want to support our mission and in return get a great season of premium TV all about hunting, fishing, foraging, and creating amazing wild food meals, check out season 1 of WildFed. You can still use the coupon code HUNKERDOWN for 25% off. States are starting to open up, and we probably won't keep this coupon code in place much longer. So if you've been meaning to pull the trigger, now's the time to act. Go over to wild-fed.com to see episode 1 and to get season 1 access. And if you know someone who might like the show, please share the link for episode 1. Again, it's free on our website, wild-fed.com. I'm Daniel Vitalis, and you're listening to the WildFed Podcast, a show about reconnecting with nature through hunting, fishing, foraging, and food. WildFed. Food is all around you. Today's guest is Professor Vincent Racaniello, Ph.D. He's a Higgins professor at the Department of Microbiology and Immunology at Columbia University's College of Physicians and Surgeons. He's the co-director of Parasites Without Borders, a global nonprofit organization that focuses on those suffering from parasitic diseases in tropical environments. Vincent's also the creator of Microbe TV, where he hosts numerous podcasts, including This Week in Virology. He puts out an incredible amount of education on viruses online through his podcast, blog, and his YouTube channel. An esteemed member of the scientific community, he's been doing laboratory research on viruses since 1975 and has received many awards for his work. I reached out to Vincent because I needed to get a solid baseline education on viruses. For years, I'd been a bit confused about what viruses actually are. For instance, how does science classify them? Are they grouped with other biota, or are they on their own as some kind of strange non-life form? Did they emerge after cellular life or before it? With so many voices shouting for attention in the online space amidst this pandemic, it was important to me to talk to someone who isn't inclined to the attention-seeking behavior I'm seeing all around me. Sensationalism seems to be spreading faster than the virus itself, from politicians, mainstream media pundits, alternative and online media personalities, and especially I hate to call them this, but conspiracy theorists. You know the ones I'm talking about. I've been particularly troubled by the folks claiming that this virus doesn't really exist, or even stranger, that viruses themselves don't exist. So the first part of this conversation is about viruses and viral ecology. What are viruses? How do they work? And what do we know about their role in the ecosystem? Are they ever beneficial? And it turns out they just might be, and while the idea of the human virome is still many years behind our understanding of the human microbiome, there's some promising work pointing to the positive roles that some viruses might play in our health and physiology. The second part of this interview is Vincent's thoughts and opinions about the current SARS-CoV-2 pandemic, the virus that causes COVID-19. I want to be fully clear. I don't agree with all of Vincent's points of view politically, but I believe in giving the floor to my guests to express themselves, and I sincerely wanted to know his opinions. For instance, when it comes to the origin story of this virus, I think there are real reasons to question the role that the Wuhan Biomedical Lab may have played in this outbreak. However, I have absolutely no background in virology and zero knowledge of what a lab like this and its protocols would look like. Vincent does, so his thoughts and opinions, even if I'm not fully swayed by his position, help me to create a much sounder worldview. I really want to get a broad range of opinions, and it would be a grievous oversight to not speak with someone with his credentials. His laboratory research on viruses make his voice very important to the conversation and to my overall understanding of the landscape of this pandemic. So get ready for a very reasonable and seasoned voice on a topic that's on everyone's mind right now. I think you'll find his enthusiasm for viruses quite compelling and a welcome counterbalance to the sensationalism that's currently masquerading itself as real news. If we can set aside our current fears, there's a real fascination to be found in the wonderful elegance of viruses themselves. Vincent really embodies that fascination. It's why his nickname is Earth's Virologist. Well, I am here with Vincent Racaniello. You are a professor at the Department of Microbiology and Immunology at Columbia's University College of Physicians and Surgeons. Welcome to the show, and thanks so much for taking the time today.
My pleasure, Daniel. Good to be here.
Hey, tell us a little bit more about your professional background and what you do and how you got, from what I can tell from kind of consuming a bit of the media. You put out a lot of media. Wow. Consuming a bit of that, you have a real excitement for viruses, which is pretty contagious.
It's infectious indeed. Well, I've been working on viruses for over 40 years. In the 70s, I graduated from college. I didn't know what I wanted to do. I loved science, got a job in a lab, found out about viruses and decided I wanted to do research on them. And so I got a PhD, I did a postdoc, and in 1982, I started my lab at Columbia. I've been there ever since, working on a whole bunch of different viruses, and I guess building a love for them. People tell me I'm morbid to say I love viruses, but I do. I think they're amazing. And then about 20 years ago, got involved in writing a virology textbook. And from that, I decided to start a blog. And then I decided to start the first podcast, This Week in Virology, which did very well. And then since then, I think we've had 6 other science podcasts, all with different hosts.
But they're all under one umbrella.
Yeah, they're one network, which we call Microbe TV. And then I started teaching a virology course at Columbia, and 10 years ago, actually, put the videos on YouTube every year. And so I do put out a lot of content. Sometimes I wonder, what am I doing? Because I'm a one-person show. I have a lot of great hosts, but as far as the tech goes, I don't have any help. So
you're doing all that. Wow.
You have to love it to do it. And
Right.
I taught myself how to do everything, but there's some days I just like to focus on content, so. But here we are. And today, now with this current pandemic, suddenly everyone wants to know about viruses, and we're very much in demand, which is great. I'm happy to keep teaching the world about virology. I call myself Earth's Virology Professor.
I was thinking about what you just said, because I've known a lot of firefighters, and while of course they never want to see people injured and property destroyed, they get pretty excited to be around fires because that's what they love to do, and that's what they train for. And I see that with special operators when they go to war, or these kind of things where atrocities happen, but there's a breed of people who are built for that kind of situation, and they get real excited to be in it. What's the last 5 months been like for you, and then for your colleagues? Because I imagine you're in a world where, like you said, the morbidity of it, like people don't really want to look at this stuff very much, and then all of a sudden, until they have to, and then all of a sudden they must seem real interested. So it must have been a real whirlwind, I imagine.
Yeah, in 2 ways. So first, there is the science part. So I have a virology lab, and it's one of just a few at Columbia. I'm at the medical school of Columbia, as you said. There's very few virology labs there. And so I feel like I've always been working in somewhat obscurity. And now all of a sudden, people are very interested in viruses. And I can't tell you how many labs at Columbia now are going to start working on either the virus or its clinical effects. And so we have started working on this SARS-CoV-2 ourselves. So I see a lot of new people entering the field, and that's fine. I think the more science that get involved, but the problem is not everyone knows about viruses, and starting with how to be safe working with them. And so that's an important thing you have to teach people. And then the other side is the communication part. So in January, I started my this semester's undergraduate virology course. And I remember the first lecture, which was January 17th, I said to the class, 120 students, who has heard of this new coronavirus in China? And 3 people raised their hands, right?
Oh, wow.
And so in every lecture, twice a week, just before the main lecture, I'd put up the epi chart of all the cases, and I would talk a little bit about it, and it got more and more intense. And then all of a sudden, school was canceled. Oh my gosh,
Right.
I said to them, can you imagine a virology course is canceled because of a virus outbreak? This is unprecedented.
Right.
And so we have been
Right.
doing This Week in Virology. Every episode, except one since January, has been on the coronavirus. And our downloads have skyrocketed. I'm getting over 100 emails a day. Now, one of the parts of our show that I always liked is we answer questions. And people have always sent in a lot of good questions, but now I can't handle 100, 150 email a day. I don't even know how to arrange them. So it's
Mhm.
overwhelming. But the thing is, they're really good questions. I feel badly, and I almost want to just do one every day to try and answer them
Right.
all.
Right. So I would imagine, like, yeah, if you wouldn't be accused of being, brash or callous, I mean, I imagine it's been exciting in your field. And then you guys must be looking at a lot of underfunded projects becoming funded moving forward, like
Yeah,
once this settles a bit.
as you know already, 800 million has been put into the NIH CDC budgets, and part of that will go to research, which is good. So, I mean, unfortunately, it's after the horse has run from the barn, right?
Right. Right. Of course.
And afterwards, it always costs more than if you had done it before. But if we had taken a bit of this money and worked on these viruses more intently, I really do think we could have prevented this whole outbreak. But going forward, I think there will be more research on these viruses. And I think we also need prevention plans. We need pandemic prevention plans. And we need a different model for making medicines that keep us healthy. Right now, all of our medicines are pretty much made by for-profit companies, and they don't want to do anything unless there's something money to be made. And that's why we're in this problem. There were no coronaviruses a threat to the world that we knew of. There were in bats for sure, but so no one would spend any money to make an antiviral. I'm convinced we could have had an antiviral ready to take care of this virus, but there was no interest. So I'm hoping now, and I see some evidence of this slowly, some new models for funding development of drugs, nonprofit based models where profit is not involved. And so maybe that will help us, because there will be a next one. Guaranteed there will be another pandemic, and we have to be better prepared for it than we were for this one.
I'd like to circle back. I kind of want to conduct this interview in a couple parts because I really want to make sure. I think what's going on right now is that a lot of people in my peer group are undereducated about virology. Most of the lay people of the world are. But now we're all talking about it, and we're picking up a lot of vocabulary from media and from the handful of experts we keep seeing. And so we're talking about something and even using the language of it, but not necessarily really understanding it. So I really want to go through sort of some virology 101 with you. And then I'd like to circle back to some of this policy stuff and get a sense of how you think it's being handled, what can be done, and what your projections are. So if that works for you, I'd kind of like to go at it like that. And
Mhm.
then one other reason is I feel bad to bring this up to you, but I'm going to because I just feel like I have to address it because I've seen so much of this. Do you remember, or were you privy to about a year ago when there was this meme going around about the Earth being flat and
Mm.
all these people buying into this flat Earth theory again? Do
Yes.
you remember that?
Yes, I do.
So I'm seeing this now with the germ theory and a lot of people who are starting to revert back to pre-germ theory beliefs amid this pandemic. And I see so much misinformation going around, and a lot of people I like and respect as friends who are buying into this meme that viruses are not actually real and that are not a causative agent in disease. So have you been encountering any of that? And could you sort of gently address folks who, because what I'm worried about is people listening to this going like, oh, these two are bought and paid for by those people who believe in germs, and I want to kind of address it because there's so much of that happening right now.
I have to say at the outset, nobody has bought me. Okay. I don't work for any company. My podcast company doesn't make any money. We have no ads. We have very little support.
Oh, wow.
I'm not in this to make money.
Labor of love.
I give away my lectures. My university said I can put them up. I don't monetize them on YouTube. I don't monetize anything. All right. And even if I had 1 million views per video, I wouldn't monetize it. I'd be leaving money on the table, as you know. It's not what I'm doing this for. So when people accuse me of being bought, and they have in the past, they're just not doing their homework. They're wrong. Okay, let's start somewhere else. So I'm doing this for love of the subject, and I want everybody to understand viruses. I have this philosophy that knowledge should be available to everyone. You shouldn't have to pay for it. And, that's hard to do. So I make my lectures available. Anyone in the world can listen to them. But yes, there's a lot of misinformation out there. However, viruses do exist. They are the most plentiful entities on the planet. All right. There's more viruses than anything else. Maybe grains of sand on the beach, but that doesn't count, right? Because they're not biological entities. More viruses than anything else, more genetic diversity. They drive this planet. There's no part of the ecology that isn't driven by viruses. And I think people have a hard time with that because you can't see them, right? You can see animals and plants and insects, but you can't see viruses. And I always like to tell people at the onset, there are just in the waters of the earth, there are 10 to the 30th virus particles. And most people can't wrap their head around 10 to the 30th. Neither can I. So I put it another way. If you took all those viruses and put them end to end, if you could, they would go 100 million light years into space. That's a bloody long way.
What?
Okay.
Wow.
And this is for something you can't even see. So that's how many there are. And they infect every living thing on the planet. The ones in the ocean are incredibly important for nutrient recycling. We didn't realize how important they are in nutrient recycling and geo cycles of like carbon and phosphorus and sulfur. Viruses are intimately involved.
Okay. So they have a role in ecology outside of just parasitism.
Huge. I mean, they are parasites, of course, but they have huge roles in ecology. For example, whenever you get an ocean bloom of an algae of some kind, the viruses always go in and kill it, and that's why they go away. And so they're very important.
No kidding.
And there are people who work on that. And so I think it's fair to conclude that most of the viruses on the planet are not harmful. And in fact, they probably are beneficial. In fact, I just told someone this morning, I think if you could take all the viruses off the planet somehow, the life would probably end in a few months because I think we depend on them that much.
When you say that they are the most abundant, are we talking about biomass or as individuals?
Just as particle numbers.
Is a viral particle a way of saying one individual virus?
Yeah. So many people equate viruses with virus particles. I think that's fair enough. Yeah. So just by numbers, they're more than anything else. But by mass, they're not that much. I mean, you can do the numbers. Take those 10 to the 30th particles. We know roughly how much each one weighs. The total mass is about 1,000 times more than all the elephants on Earth. Okay.
That's cool. What could you define a virus particle? Because I feel like you're going, yeah, that kind of works. But like, what does it really? What do you mean when you say particle?
So particle is as the word says, something particulate, right? It could be a grain of sand, except it's a lot smaller. It just has substance to it. But what a virus is, it just happens to be so small that you can't see them. So we can't imagine them as particles, but we in the field call them particles. But they're basically assemblies of molecules, and they typically have some kind of genetic information. It can be DNA like ours, or it can be RNA, which is unusual. In us, RNA is in the form of messenger RNA and that sort of thing. But in viruses, it can be their genome, which is quite interesting.
For people listening, so DNA would be like the blueprint for biology, but RNA would be sort of used to code out proteins,
Yeah, that's a good
something
way to
like that.
put it. Whereas in some viruses, the RNA is actually the blueprint, and why that is
Okay.
very interesting. But um, so it's minimum, a piece of RNA or DNA, and it can be very small. It can just code for one or two proteins, or it can code for thousands of proteins. They range in size. And then it's usually wrapped in some kind of protective coating. It can be a protein or it can be a membrane of some kind. And that's why we call it a particle. All right. So if you could zoom in on these with an electron microscope, you could see they're very distinctive particulate things. And
Okay, but you don't use the word particle because the individual virus unit's been broken up in any way. An intact
no, no.
individual virus we would refer to as a particle.
Yeah, we do.
I was Okay, got it. I was seeing in your lecture, I wanted to make sure I understood that. I was seeing in your lecture you were talking about how over the years that you've been teaching, you have rewritten your definition many times
Right.
for virus. I thought that was fascinating that there isn't a consensus about how to define them. They seem like they defy our neat categories. So the if I correct me if I'm wrong, but your 2020 definition is an infectious obligate intracellular parasite comprising genetic material, DNA or RNA,
Hmm.
often surrounded by a protein coat, sometimes a membrane. So that's sort of your current
Yeah,
way of looking at it.
that's right. And so one of the things so what never changes is obligate intracellular parasite, which means these viruses have to get inside of a cell to make more viruses because they can't do anything on their own. They're incapable. They don't have any of the machinery that they need. They have some instructions to reprogram cells to make viruses, but without a cell, they're useless.
So when they have a protein coat or a membrane, if I found an individual particle in the environment, its membrane would have been manufactured by another organism because it doesn't have the organelles to manufacture itself, even
That's
its
right.
own parts,
That's
correct?
right.
Wow,
Everything
that's
is
fascinating.
everything's manufactured by the cell. Absolutely.
What are they though? Like, I understand the definition, but and I have the cliff notes because I went through your lecture, but for people who are I know this is the age old question in your field, but are these alive, and which comes first, cellular life forms or viruses?
So, are they alive? It really depends. If we're talking about these particles, right? These pieces of genetic information wrapped in protein or membrane, those aren't alive. They have no way of living, or they have no way of multiplying on their own. So I don't see them as living. But once they get in the cell, they take over the cell, and now it's a virus infected cell, and that's certainly alive. So I look at it
The
as
cell.
the cell is alive, right? Until the virus
Yep.
kills it, of
Right.
course. So I look at
Right.
it as an organism with two phases. You have this particle, which you could almost think of as a spore if you wanted to, which doesn't do anything until it gets into a cell. And then the cell is a living virus producing cell. So in that phase, it's living. But you asked what came first, the virus or the cell? Well, that's a very interesting question. And we're starting to get more and more information on that. And the current hypothesis is that on Earth in the early days, billions of years ago, when the Earth cooled and organic molecules began to form, we think there were what we call replicators. That sounds very sci-fi. But
It
replicators,
does, yeah.
which was just small molecules of RNA, actually, that formed spontaneously, and they could actually copy themselves. There were no cells, there were no proteins, but they had the ability to copy themselves. And then they existed for many years. This was called the RNA world because that's all there was. And then
Wow.
proteins evolved, and then DNA evolved. And then when DNA emerged, and I'm skipping many, many years here, then you could make cells because cells require a lot of genetic information that's not possible with an RNA. So now we have these primitive cells emerging. This is a billion years ago. And these little self-replicating replicators, they are, I'm going in that cell. It's a lot nicer in there than out here. So they go in the cell and they find a great environment for producing more of themselves. And then they begin to steal proteins from the cell and build themselves a home. and they build themselves particles. So all the proteins that surround viruses come from cells. They're stolen genes. And so those viruses pick up a protein shell and they leave the cell and they go to another cell. And that's the birth of viruses. So that's how it's generally seen.
It's, yeah, I'm sure that that's a very simplified version of it, but it begs the question for me: how does the forensics work? Like, is there anything like a artifact, like fossil type evidence, or is it all done through, I guess, computational biology stuff? Is it all DNA evidence or what we see, residually in genomes?
Yeah.
Like, how do we know about past viruses?
Yeah, so there are no fossils in the sense of bones, right? Like we have for dinosaurs. The oldest virus that we actually have is just 30,000 years old. It was recovered from a frozen Siberian ice core a couple of years
Oh,
ago.
wow.
And, we know how old those ice cores are. So that's how we know how old the virus is. But as you said, we can use computational methods. And so one of the really enabling technologies in the last 20 years has been to be able to determine the base sequences of these genomes, of the genetic information, the 4 bases, A, C, T, G. What's the order? And we can do massive sequencing projects. We could take the ocean and sequence everything in it, for example, readily. And so we have now many, many sequences of different viruses and their hosts. We can relate the hosts on a phylogenetic tree of life, if you will, by how they're related to one another. We can fit the viruses into that, and we can begin to see very faint signatures. If we look at the viral proteins, the ones that make the shell, we can see very faint signatures of them in the cell protein. And that's why we believe that all these viral proteins came from the cell to begin with. Now, the caveat here, of course, is that we're looking at everything that's around today and we're making extensions as to what might have happened a billion years ago. And that, yes, you can get artifacts because it's, we would love to have if people often say to me, if you could do anything, what would you do? And I say, I would take a time machine back a billion years and take samples on the way back and see what was there, because that would answer so many questions. Because you can imagine that what was around then is so different than what's around now that we wouldn't recognize it. And so that's part of the problem with these methods. But, when you accept that they're not perfect, these are the kind of scenarios. And you come up with scenarios that are more likely than others. You could come up with 2 or 3 scenarios for how viruses emerge. But the one I told you is the least complicated and requires the least number of assumptions.
All right, got it. Occam's razor. Um, yeah. So you're kind of having to look at shadows and try to imagine the 3-dimensional object that is casting it in a sense.
In a way, yes.
Yeah.
I mean, we can do more with today's stuff. Okay, I don't want you to think that the whole field is like that.
Right. Of course, I got you. Of course. Where do they fit in this like phylogenetic tree of life, and how are they classified? Like, what's the convention for scientific naming? And do they have the way like many mammals would have a trinomial name to designate them as a subspecies? Do they ever have infraspecific epithets and, subspeciation, or how is that? How are they named? Like, what's the convention?
So there is you ask how they fit in the tree of life. Well, they don't actually. Most people don't put them in the tree of life, although they should be, because they have evolved alongside of living things, right? So, but we have a separate tree of viruses where we can make a tree based on all the genome sequences that we have. We used to do it by physical properties, just like we used to classify and make trees of animal life by looking at them. Now we do it all by genetics, and the computer does it for you, and it seems to be more accurate. So we have trees for viruses, and they're arranged in the typical classification, kingdom, phylum, class, order, genus, and species. So many viruses are part of a genus, and then there's species. There are families that collect genera and so forth, and higher and higher. And there's a naming committee that names viruses officially. And if you discover a virus and you want to give it a name, you have to make an application to this committee, and they will consider it.
Right. Do we have when we like can we see viruses with scanning electron micrographs and as it relates to or other technology, or are we when I'm seeing images, like we're all, I guess inculcated now into the COVID-19 or the novel coronavirus, we've all seen this image over and over again. Do we get to actually see that, or are these assumptions based on, because what I hear a lot about is sequencing. So that leads me, just as a layperson, to think that we're finding, sequences, but we're not necessarily seeing the particles themselves. So when do we see those particles, or how do we see them? And after sequencing, like how do we go from finding a virus and sequencing it to actually being able to see it, or can we?
So we can see them. In fact, electron microscopes have become more and more powerful. And now you can readily see virus particles. In fact, the new corona back in January, when it was first discovered, they did some electron micrographs and they could see the pictures and say, oh, this looks like a coronavirus, because every kind of virus has its own unique morphology. But we can do even better. Some of the beautiful pictures you've seen of SARS-CoV-2 are made by using an electron microscope to take pictures. It's kind of like a CAT scan. You take many pictures of virus particles, and they're all the particles are in a different orientation, right?
Yeah.
And then you can use a computer to assemble them all into a three-dimensional view,
Right.
and you can
Build it
get
up.
amazing structures. And that's some of the structures that you're seeing of these viruses. So we can know in very, very precise detail. In fact, in some cases, we know at the atomic level where every atom is in a virus particle. So it's really based on
Really?
evidence. It's not just
Including in
extrapolating.
its membrane and everything?
So the membrane can be modeled by electron microscopic techniques. The proteins are more suited to that because they're more ordered. A membrane is fluid and harder to
Would the
pin down.
membrane be like a phospholipid membrane?
Yes, it would be a phospholipid bilayer, right? 2 layers.
Wow. So fairly complex.
It's just the same as our cell membrane. It's taken from the cell as the
Wow.
virus leaves. And so these coronaviruses that you see, they're all little bits of cell membranes taken from someone somewhere. They're stolen. That's why they're parasites, right?
What are they taken in the way that I might take your wool sweater, or are they taken in the way that I might take your sheep and shear it and then build a sweater out of the wool that I took?
I don't think either one works. The viruses get inside the cell. It's more like a Trojan horse thing, right? Where you see this
Right.
nice horse and you don't know what's inside it. So a virus gets inside of a cell. The virus basically disappears from view. And then over the next hours, it's inside that cell, redirecting all of the cell machinery to make new virus particles.
To make it, yeah, including those membranes.
Well, then these coronaviruses or any virus with a membrane will then leave the cell. One way of getting a membrane is to actually leave the cell surface. We call it budding. Little blips form and then they pinch off, and that's a new
No
virus
way.
particle. Yeah.
No
So it's
way.
stealing at the very last part of the cycle a little bit of membrane. And that stealing can happen at the outer membrane, but it can also happen within. There are lots of membranes in cells that can happen from also.
Man, it's hard to you mentioned in your lecture the sort of risks of anthropomorphizing
Mm-hmm.
these viruses, but it's so tempting. I feel the temptation right now, and
Yeah.
because
Yeah.
there's something that's, to my human mind, so elegant and sophisticated and intelligent about this. So
Right. And
it
that's
must
exactly
be,
why
is it
you shouldn't. That's exactly why you shouldn't, because you don't know anything about what's going on there, and to ascribe human qualities to it is misleading. That's why I don't want people to anthropomorphize.
right? Under optimal conditions for a virus, and I'm sure, I mean, I'm sure as we go through the taxonomy, there'd be a tremendous amount of variation, but under optimal conditions, how long can they remain? I don't know what the word would be. Is it dormant or viable? How long is a particle viable before it gets, into a cell? I
Mhm.
guess, what are the longest lived ones that we know about?
So viable is a good word, or infectious. So infectious means that
Okay.
this particle can infect the cell and reproduce. And the 30,000, well, that one in the 30,000-year-old ice core, that was frozen and it lasted for 30,000 years. So
What, it was infectious?
yeah, they thawed the ice and they put it in the lab and it infected
Wow.
cells. Yeah.
Wow.
And
That's
so
incredible.
in a freezer,
So we don't know.
freezer
Yeah.
well, so I would say 30,000 years is the longest we know. But I have freezers in my lab full of viruses, and they can last a long time. So when you freeze a virus, it keeps it indefinitely, essentially. As long as you have
Yeah.
electrical power, the virus will
Wow.
stay.
And they're most susceptible to, I would imagine, heat, light, oxygen, UV, things like that. Like what are the
Hmm.
things that really break them down in the environment?
So heat is one, UV light is another, um, low pH is another one that
Oh,
viruses
okay.
don't like. It depends on the virus, of course. Some viruses that
Yeah.
that you ingest and would go through your stomach, they're quite resistant to low pH. So poliovirus, I can put it pH 2 in my lab and it won't blink. It
Wow.
doesn't lose
Wow.
infectivity.
pH 2 is pretty low. Wow.
And some viruses, if let's say on the surface of my desk, a coronavirus might last a day, but a norovirus, which causes an intestinal infection, could last a year, could remain infectious for a year. So there's a
Wow.
huge range.
Wow. There's also quite a lot of range in the morphology. And you were mentioning before that we're using gene sequencing for the taxonomy now, right? So it
Right,
wouldn't
right.
be morphological, but looking at, like, for instance, are they called bacteriophages, which have that almost like a looks like a spider with a crystalline
Right.
abdomen type shape, right?
Yes.
So you've got this from these very geometric complex structures to, I'd say with the novel coronavirus, looks a little bit like a Nerf toy or something, a little silly compared to some of the ones that I've seen. So talk a little bit about, I guess, the range of morphologies that we see. And then when you group them genomically, do they tend to group out a bit morphologically anyway?
No, that's a good point. So we used to use morphology as a way to classify viruses, but we don't anymore because the genome just usurps that. And it turns out that the genome will lump together viruses with or without membranes. It doesn't really matter because
Oh,
that
wow.
tells you their origins. They evolved from
Hmm.
common ancestors. But there are not a lot of different kinds of virus particles. There are, I would say, the kind like the coronaviruses, they have a membrane, and those would be like coronaviruses, and um, like Zika virus would have a membrane. Influenza viruses have a membrane. Measles, they'll have this membrane within which is some nucleic acid, and then in the membrane are proteins, which we call spikes, and they're very important for attaching to cells. So,
Is that what we're seeing all over the surface of the,
yeah,
you're calling it SARS-CoV-2?
SARS-CoV-2 is the official name of the virus, right?
SARS-CoV-2. Okay.
Yeah, and
So is that what we're seeing, those spikes? And are
that's
they
right.
called the S spike proteins?
Yeah, S is for spike, right?
Okay.
It's an abbreviation. They have slightly different names depending on the virus, but the generic name is a spike. But for coronas, that's actually the real name, spike protein, as well. But
Okay.
for influenza, you would say it has a spike, but it's got a different name. It doesn't really matter.
Okay.
But those range in size. They can be small, they can be large. They all have a similar appearance. They have a membrane, they have spikes. So that's kind of one class of virus. Then there's another class with no membrane, which is made of pure protein shell. And these look symmetrical if you look at electron micrographs of them. And viruses like norovirus and poliovirus are in that category. And
These will have that like pill shape or that kind of like bacterial shape.
so if you think of the bacteriophage you were talking about it's got the legs. It looks like it has legs and a
Yeah.
neck, and then the
Right.
head looks like a icosahedron, and that's in fact what
Yeah.
it is. So a
Yeah.
lot of viruses are just made of that icosahedral head, and they don't have any spikes in their
Oh,
shell.
okay.
They're just pure protein around a piece of nucleic acid. And so those are the two big categories of viruses. Then we have another set which don't fit in any of those. They're more complicated and they can be really big. And these are like the Siberian 30,000-year-old virus wouldn't fit into any of these categories. They're really huge and don't have any symmetry whatsoever.
Huge on what scale exactly? Like, give us some sense of scale for these things.
The smallest viruses, so I have to use nanometers as a measure of this. The smallest viruses are about 20 nanometers in diameter. Okay, a nanometer, is a millionth of a meter,
Okay.
and they can be
Is
as
there an object we could compare it that could we, like maybe the width of a hair or something. Any idea how
Oh,
many
it's much
nanometers that
smaller
would
than the
be?
width. You could put hundreds of these in the width of a hair.
Okay, that's what I'm looking
Or
for.
how about the head
Like
of
how
a
many
pin?
nanometers is a
You could put
right? Okay, give us a head of a pin.
head
There
of
we
a
go.
pin. You
That's
could
useful.
put
Yeah.
thousands of 20 nanometer particles on the head of a pin.
Wow. Okay. Right.
And then there are viruses that are 1,500 nanometers in diameter. They're huge, relatively
Wow.
speaking, to the small ones. That's why I say huge. And those
Hmm.
you can just see in a light microscope. So in a light microscope, you can
Oh
easily see
wow.
bacteria and yeast, right?
Really?
And these
Oh,
viruses are so big you can actually see them. Yeah. So that's a big
interesting.
range, and those bigger ones are the ones with the morphology that doesn't really fit anything that I've told you about.
One of the questions I had written down is, are they ever symbiotic? And then I realized, well, that's actually not the correct term, I imagine, because they're not biotic, right? But are they ever symbiotic? What are some positive roles that viruses play in ecosystems? You mentioned before, helping to turn over the blooms of algae. Are there other areas where we can look at them? And I guess, like, for instance, when we look at foods, we know that bacteria play a tremendous role in lots of foods that
Mm,
we've enjoyed for culturally for thousands of years. Well, culturally, literally culturing foods. So, cheeses would be an
right,
example, and
right.
wines would be an example where we have fungi playing a role. Are there any foods where viruses play a role, and are there just general symbiotic roles that they play with other organisms? Hey, we'll get right back to the show in a moment, but first I wanted to tell you about Paleo Valley's 100% grass-fed beef sticks. I recently received a package in the mail with all of their flavors. And by the way, I like them all pretty equally, but I'm partial to the original flavor. Anyway, they were interested in sponsoring the show, and I wanted to test out their products first. Now, I already knew the benefits of grass-fed, grass-finished meats with their substantially higher content of omega-3 fatty acids, conjugated linoleic acid, and glutathione, but I hadn't had many meat snacks that I really liked. Well, I still haven't. And that's because my wife likes them so much that she's got to most of them before I could. Seriously though, these meat sticks are awesome and perfect for your backpack, when you're traveling, or just need a high-protein, low-carb snack. If you're doing a paleo, keto, or carnivore diet, or just want a quality meat source on the go, visit paleovalley.com/wildfed. Again, paleovalley.com/wildfed. That'll get you 15% off your order, in addition to the bulk discounts they offer right there on the site. Again, go to paleovalley.com/wildfed and your 15% off will automatically be applied at checkout. Again, paleovalley.com/wildfed. There are several flavors to choose from, and they're all excellent. These are not like the spam-tasting meat sticks from that highly processed brand at the health food store. You know who I'm talking about. These taste more like a summer sausage made love to a Slim Jim. And as I mentioned, they're Avani approved. Trust me, she's picky. Again, go to paleovalley.com/wildfed and get 15% off your order. Now, back to the show.
Well, all those uses of bacteria that you just cited, many of those properties of bacteria were acquired by viruses bringing the genes into the bacteria. So they're really good at transferring genes between different bacteria. And many of those strains can be virus infected, and that gives them some of their ability to make interesting wines and cheeses and so forth
Wow. Okay.
at just one level. And then there is another level where we actually think viruses are beneficial. So the clearing of the bloom is good for us, but for the bloom algae, it's not good for them, obviously, because the
Right.
virus is killing them.
Right. Right.
But there's a lot of turnover of organic material in the oceans. There's something like 10 to the 20th infections per second, virus infections per second that go on in the top centimeter of the ocean. And this liberates tons and tons of organic material on a daily basis. And it's really, as I said earlier, it's really important for recycling of various nutrients, geochemical cycles. But more close to home, here's an example. Two examples that I really like. One, there are some grasses that will grow at hot temperatures, like the grasses around Yellowstone Park, around the hot waters, right? Those grasses can grow at 50 degrees centigrade, and they can grow that temperature because they have a virus in them that gives them thermotolerance,
Okay.
right? without the virus. And you can take the virus out of the plant and try and grow it. It will die at 50 Celsius. So it's
Wow.
giving
Okay.
thermotolerance. And many people feel that our modern crops, which, as you know, are monocultures, they lack resilience because they don't have the viruses that a wild plant would have in it to give them thermotolerance, drought tolerance, and other beneficial properties. And a lot of people are working on that to try and pin that down.
There's this role that you're talking about where they can infuse genetic material
Yes,
into more complex organisms, and then are they also affected in a reciprocal way? Do they end up with sequences from I don't more complex life forms that they infect?
for sure. Yes.
Wow, so it's a real mercurial role, like that sort of
Oh,
that
absolutely.
old Greek idea, Roman idea of the sort of messenger of the gods. So they're moving between organisms and they are seeding genetic material across species in a
Hmm.
sense.
Absolutely. Yeah, we call that horizontal gene transfer. But yeah, they infect the cell. They pick up a bit of its DNA. They may keep some of it. They may bring it to another cell. Yeah, they're shuffling DNA all over the place.
Wow. I would speculate that that would play some fundamental role in evolutionary history of some organisms. Like, do we know of any, are there examples of genes that have been picked up by viruses that then become functional to an organism down the road in its natural history?
Yeah, I have a great example in humans. So, humans and many other mammals have placentas, right? So they can give live birth. And it turns out that there is a protein that's essential for the formation of a placenta in all the mammals that have placentas that was delivered by a virus many millions of years ago.
Neat. Okay.
And the organism, the
Wow.
host, took this virus protein and used it to build the placenta. So, if it weren't for that virus, we'd be laying eggs.
Right, right, or some kind of mitosis. All right, so then that's really interesting. I guess for a person in your position, well, the average person hears viruses and thinks immediately their mind goes to negative places. So I imagine when you spend time looking at a lot of different viruses, because it's interesting in your definition, they're all infectious, but that doesn't mean that they all have a pathogenesis, right?
That's
So
right. That's right.
so when you think of viruses, you're not just thinking pathogen, you're like the average person might, but you're sort of thinking about the tremendous roles that they play across ecology.
I am big, and I mean we have a human centric view of viruses, right? Because they make us sick. That's how we discovered them initially. They made us sick, and our early research on viruses was all influenced by them by finding the causes of human diseases. And we ignored all the other viruses out there. But now we know, for example, that many animals, mice, rodents, bats, what you name it, plants, they're all infected with viruses and they're absolutely fine. They don't get sick. They've evolved with them a lot longer than we have. Humans are relatively new inhabitants of Earth,
Hmm.
and viruses have been around since the early days. So many other animals have had a long time to evolve with them and to be okay with their presence. We haven't, because we've acquired them recently. So for most animals, for most life forms on the planet, having a virus is not harmful. There's no disease that results. And I think in many cases, they're probably beneficial.
Could you estimate like how many unique species of virus a person might have in their body at a given time?
I would say at least a dozen different kinds of viruses in your
Okay,
body.
because I picked up a term virome from your lecture,
Right.
which I hadn't heard. Now I've interviewed quite a few people on the human microbiome, including folks who've gone to be with indigenous folks in Africa just to see the effects of
Yeah.
eating their diets and what impacts are on their microbiome did they see, and what health benefits did they see. So I find all this really fascinating, but I'd not heard the term virome. So bring people up to speed on what that means and I guess where that field might be taking us to.
So a virome is very much like the microbiome. It's the collection of all the viruses in you. And so it's like all the bacteria in you is your microbiome, or actually more specifically, it's the genome or the genetic information of all of those bacteria, the microbiome, and the virome is the genetic information of all the viruses in us, which many people are interested in studying because we think they're beneficial. There are lots of viruses that humans carry, at least a dozen, probably more, that don't seem to have any harm and are likely to be
Hmm.
beneficial. And finding out how they are beneficial is hard because, you can't do experiments with people, but
Right.
we try and use animals to sort that out. And it really does look like, for example, every pint of blood that's collected in the world from humans has got viruses in it. You can't avoid it. If you threw out your blood supply because of the presence of a virus, you wouldn't have any blood to give people. And so we've learned that some of them are
Mm.
okay. They don't harm us. And I think many of those are probably beneficial. So that whole collection of viruses, that's our virome. And you have one that's unique to different parts of your body. You have a skin virome, you have a gut virome, a lung, an eye, a brain virome. And so these things are insinuated in us. If I do one thing in all my teaching, it's to get people to understand that these are not things that are here to make you sick. Some of them happen to make you sick, but most of them are okay and probably help you.
Well, it's interesting because we're really in our infancy in understanding, the role bacteria play in our health.
Right. Right.
I mean, it's really only been in the last decade that that's become kind of a mainstream talking point. And so, how far behind bacteria, the science of the microbiome are we with viruses, do you think?
We're quite far behind because with bacteria, we can do experiments. For example, we can give people antibiotics to clear out
Oh,
the
right,
bacteria in their gut,
right.
and then we can give them back bacteria. We can give them fecal transplants, for example, and we are learning
Hmm.
that it can cure some diseases. Right? We have no way of clearing the viruses from people and doing that experiment, so it's very hard to know. So as a consequence, the virome studies are lagging far behind the microbiome studies. I mean, at some point they will catch up. They will have the technology to do what we need to do. Things like CRISPR, gene editing tools, are going to make it possible to remove specific viruses from us. So eventually we'll get there.
Because we have these broad spectrum antibiotics that are so effective, but the world of antivirals, I don't really know anything about it. So I've been hearing the conversation that's taking place, in the news sphere, basically a talk about antiviral therapeutics and such. But why are they so difficult to produce, and how is it that they work?
So they're hard because taking bacteria as a comparator, we can make antibiotics because bacteria are their own thing. They have everything they need to grow, right? They have their own machinery, and it's different enough from ours that we can make drugs that target bacteria specifically. But every virus needs to get into a cell to make more viruses. And so finding drugs that don't harm the cell is very difficult.
Mm,
You have to
okay.
focus on the virus part. And so you're limited, and even many of those. And so every virus is so different that you don't have a broad spectrum drug that will hit all viruses. And that's part of the
Okay.
problem why it's so hard. We have about 100 antiviral drugs that are licensed right now in the US, mostly against HIV, herpes viruses, and hepatitis C virus, right? Most of them for those because they're chronic lifelong infections. But for all the others, it's very hard to do.
And I guess one more question I have about viruses in general, and then I want to start talking a little bit about the current situation. But um, when viruses infect a cell, are they like a drunk burglar who smashes a window and jumps in, or are they like a master cat burglar with a set of lockpicks who specifically devises a quiet entry strategy?
Well, if you want to anthropomorphize them,
I'm not a virologist. I can get away
you
with
can do it.
it.
They are very sophisticated, absolutely not a blunt tool at all. They go in with the most amazing toolbox that subverts and takes over everything in the cell that they need to make more virus particles. And we're learning about this over the years, how it happens in individual proteins. And here's how insidious it gets. Not only does the virus make proteins that switch the host to making viruses instead of more hosts, but it actually, many viruses have evolved to utilize host proteins, which are originally designed to fight viruses, but the viruses turn it around and use them for their benefit. It's really remarkable. So This, and that's a broad stroke picture of it, but, and, not only within the cell, but, when you're infected with a virus, your body makes defensive moves. You make antibodies and cells and various proteins in defense, and viruses have evolved to counter most of those. And that's why they're still around. Our immune system is really good. In a healthy person, the immune system is fabulous, but viruses are better because they have evolved to evade most immune systems. Otherwise, they'd be gone. They would be off the
Yeah.
planet because they just can evolve so quickly, faster than anything else.
When a person recovers from a viral infection, does an individual cell ever recover? In other words, can it go from becoming a viral manufacturing machine back to normal function, or is that cell going to be lost before it could ever actually recover?
In most cases, the virus is killing the cell. I mean, it can happen quickly, it can happen in 8 hours or so, or it can take days. It depends on the virus. In very few cases, though, the virus can get in and and suddenly become silent, and the genetic material remains in the cell, but the cell lives on. And then that's the problem, in fact, with HIV, is the virus gets into some cells that are very, very long lived, and it becomes silent. And those cells are a problem because at some point in the future they can start making virus. But in general, the
Okay.
viruses do kill the cells that's why we call them parasites, because they harm the cells that they're multiplying in.
Right. Um, let's imagine that, yeah, I had Thanksgiving dinner and, boy, that turkey put me to sleep and I'm in cryogenic sleep for, 6 months. I wake up, and I have no idea what's going on. Could you give me a sit rep on real, like, biggest picture executive brief on what's going on today, with the novel coronavirus? Um, kind of, yeah, just your biggest picture overview, because I want to start working from there.
So one thing you need to know as soon as you wake up, is that every human virus we have, whether it's measles or polio or mumps or cold viruses, we got them all from animals at some point in our existence. And some of them we got thousands of years ago. We got measles from cows 15,000 years ago. And some of them we get now. And that's what this SARS-CoV-2 is. It's a brand new virus we acquired from an animal source, possibly a bat. We had the virus in the bat, which encountered a human somewhere, and that started a chain of infections. And this is so new. This virus has never been seen by humans, that everybody on the planet can be infected, and it's traveling around the world. We have wonderful transport systems that can bring you around the world in a day, and we have lots and lots of people who can serve as hosts for this virus. We have made the perfect environment for human viruses to flourish. And so that's what's
Mhm.
happened. This new virus has emerged, and it's going through humanity. It's going to be a while before things calm down, unfortunately.
If I had asked you 6 months ago how likely, a coronavirus, I mean, I can't help but note that, what was it called? Event 201, the Bill and Melinda Gates Foundation and, John Hopkins were working on a coronavirus simulation in November. So,
Mhm.
obviously, there was people in the world who had thought about the possibility of a coronavirus infection or pandemic. Um, was this on your radar at all, the possibility that, in particular, a coronavirus could, emerge like this, and become so infectious?
Well, those of us who work in virology know that coronaviruses have what we call pandemic potential, right? Because
Okay.
we've known that there was a SARS-1 in 2003 where the virus went to many countries. It didn't, it only infected 8,000 people. So obviously it has the potential these viruses have the potential. And that woke us up, because we didn't realize that there were lots of coronaviruses in bats in many countries in the world, not just China. And people started to look for them and found that, yes, there are SARS-like coronaviruses in bats. So they are a threat. So we have always known that coronaviruses are a threat for a pandemic. And, people have been trying to get funding agencies interested in putting money up for this. It's very hard to get research for something when it's not an issue. And so
Right.
that has been the problem. And that's why we're not ready. But we have known, and it's not just coronaviruses that are pandemic threats. There are many other viruses out there that are threats, but now this one has come. And I'm actually surprised that it has spread this far, because the last SARS outbreak was limited, but this is a very different virus. And so I'm actually surprised. I didn't think it would spread this much, but in hindsight,
Oh, that I'm curious about that. So when you first started, I imagine you heard about it early. Did you hear about it in November or December?
I heard about it in December. And they had just seen this outbreak of unusual pneumonia in China of like 40 people in Wuhan. And then they said it was a coronavirus. And I said, well, that's interesting. It's a SARS like corona. Well, the other SARS died out. We were able to contain it. So probably this will be the same. And that was my mistake. I shouldn't have said that. I should have said, we don't know if it's the same or it's different. And it turned out to be different enough that we could not stop it.
Was that a general feeling for, at least here in North America? Did a lot of virologists feel that? Were a lot of your colleagues thinking the same thing? Because I think a lot of lay people were thinking that because, and I'll ask you about the media response later, but our press, obviously their business model is based around sensationalism. It's how they make money. So it's understandable that they really love to make everything a crisis.
Exactly.
So I think there had been so much crying wolf going on that when I started to hear, hey, there's a wolf running around, I was like, yeah, well, and then, it slowly started to become obvious this was something different. But was that a general feeling in your world?
I think it depends. I mean, I think the coronavirus virologists, and there are not a lot of them, but there are some, they were worried because they knew what happened with SARS. And we have this other virus, MERS coronavirus, which is circulating in the Arabian Peninsula. It's been for years now, slowly percolating there. And so I think a number of virologists were worried about it, and then there were some who were not. I definitely remember speaking with some virologists who thought, no, I don't think this is going to spread globally. It's probably going to be restricted relatively quickly. China will shut down and that'll be the end of it. So I think it was mixed, as with anything else that you would expect.
What do we know about? I know the numbers have been changing a lot, and I get frustrated a little bit with. There seems to be expectations that all the testing should be ready, or expectations that we should have the correct numbers, and it seems like, hey, we're in a very fluid situation. So it's understandable to me that we've seen, for instance, the case fatality number change so much. I mean, that seems like, of course, until we have hindsight. But what's the sort of current thinking right now, or what's your current thinking about the numbers, the R naught, the case fatality rate, things like that? Or where do you think those numbers will land? Once we've had more time to analyze all the data.
All right. So R naught, the transmissibility, right? So that's the average number of people that an infected person can infect. This was determined very early on in the outbreak in China, and it was estimated to be between 2 and 3. So an infected person could infect, on average, 2 or 3 other people. And obviously, since it's an average, you can have more or less depending on the infected person. I think that's correct. And now we've done intervening, which it makes it harder to determine the R naught, because interventions can lower it. And if you lower it to 1 or below, then you can stop the outbreak.
Okay, so this is the social distancing, isolation, quarantines, all of these things that mitigate.
Now, the testing is a problem because you can't test everyone, just too many tests and not enough labs to do it. And the U.S. got started late on testing, and so we are vastly underestimating the number of infections. And in China, also, they got overwhelmed, right? They were cranking out 50,000 tests a day, and you can't sustain that, especially in a country with 1 billion people. It's just impossible. So I suspect there are far more infections than we know. And one thing that we do know about this virus is that 80% of human infections are pretty mild, and you might not even recognize that you have some infection. You may have a sore throat or just a mild cough. So I think there are a lot of people infected who don't know it, and we're not picking up anywhere near them in our testing efforts. If we could test every person in the U.S., then we would know, but we can't. We can't do that. So I think the numbers are at least tenfold underestimated. You see 2 million, 2.5 million confirmed infections globally, but I think it's more like 25 or so. And that's good, right? Because we want to have more people
very good.
infected and immune. And I'm hoping that, we're starting to come out with serological tests, which will measure, proteins in your blood that are made to block the virus. We can measure those, and then we can get an estimate of how much of the population was actually infected. And so you can go into a community and say, take a few hundred people and look in them and go, oh, 50% of this community has been infected. That's pretty good. More than we thought, but based on the diagnostic tests.
Today's April 21st, 2020. And at this point, we're hearing a lot more about, there's, I think, 3 states getting ready to open up next week. And there's a lot of discussion on both sort of ends of the spectrum about what should be done next and what should be the criteria for opening up. And one of the big things we keep hearing is about antibody testing. Maybe more people are infected and are asymptomatic, which might make the actual case fatality much lower. And I think a lot of people are starting to wonder, because I'm sitting here at home wondering, what if I have already been infected and I'm asymptomatic and I am recovered and I could go about my day? It would be a real bummer to languish, here in economic depression. So I'd like to get busy, So what was your thinking on that? Like if you were in control of this rollout, I guess maybe actually I'm jumping ahead too. I want to talk a little bit more about this idea, what it would mean if more people were infected, but asymptomatic. And then I'd love to hear about sort of from day one, how your dream scenario, how you would have rolled it out. And then what actually was done, and where you think we should go from here. But go, that's a lot of stuff. So let's pick that apart. If more people are asymptomatic carriers, that would mean that this was a lot less dangerous than was initially thought, right? Do you think that there was an overreaction, or do you think this was a correct reaction because we didn't know?
Oh, that's a tough question, because what we're doing is the 20% that are serious infections, right? Those people, many of them have to go to hospital, and the hospitals only have so much capacity, right? They only have so much surge, and they're going to get overwhelmed, and then people die. So we wanted to avoid that, and that's why we're flattening the curve and trying to keep the total number of infections down. Now, the downside of that is that less of the population is being infected because we're not encountering each other. And why is that important? Well, the R naught, 2 to 3 will keep the outbreak going. But if the population is immune, you can lower the R naught. And in fact, if you have an R naught of 2, then you need half of the population to be immune to infection to stop the outbreak. If you have an R naught of 3, it would have to be 70%. And so that's why we'd like to have more people infected and protected, because that'll spontaneously stop the outbreak. The virus will not find hosts to infect when 50 or 70% of the population is immune. At this point, we don't know what fraction has been infected. We have no idea. And it's being suggested that these antibody tests, which are being rolled out, are going to tell us this. They're not going to be able to deploy them on big enough numbers to really give us information. Plus, they don't actually tell you if you're protected. They just tell you if you've made a response to the virus. So there's a little bit of faith in that we're hoping that they're going to be informative, but they may not. And so
there's a pretty statistically significant false negative, false positive problem as well.
Yes. These tests aren't perfect, especially when the population has low seropositivity to begin with. When few people have been infected, their accuracy is not great. And these tests are being rolled out by a number of companies, and they're not all. The federal government is not checking them all. They're just saying, "Yeah, sell them."
Right.
They don't have time; they can't handle it. And, that's part of pandemic preparedness: being ready to do this, to have an infrastructure to make tests and distribute them. We didn't have it; we had no infrastructure. So that's where we are. And going back, so look, we cannot stay closed forever. It's just we can't trash the world economically and socially, right? People are out of work. People are getting messed up in their heads. I totally get that. So we have to go back to work at some point, and with that, the chance that we're going to get infected. Now, I think if we delay until the end of May, June, the weather will change sufficiently so that'll impinge on transmission, and it will be less of an issue. And
Due to
then
increased ultraviolet light?
increased sunlight and also humidity. As the humidity rises, these droplets that bring the virus out of your nose, take on water and they fall to the ground really quickly. And
Okay.
so I think that's going to make transmission very low by June. And so at that point, I think we look at the curves and say, transmission has dropped, time to go back to work. It's likely that in October, November, we'll have another outbreak because as the weather gets colder and the humidity drops, then transmission will rise. But it will be a far less extensive outbreak because there'll be more people immune, and we'll be able to check and see who's immune by that time. And then maybe in 2021, we'll have some vaccines. So that's how I
We'll
see
also
this
have a much
carrying.
better, our medical system will be much more robust and prepared to handle that second wave than it was for this
Yeah,
first one too, right?
exactly.
So as far as the flattening the curve. Boy, there's so many little pieces here I want to ask you about. How realistic is a vaccine? I've heard some folks, again, it's like being a layperson. I know so little about it. And I hear everything from, I hear a lot of 12 to 18 months. And then I hear some folks saying, hey, there's no real guarantee that we'll be able to create a vaccine for this. So as somebody who doesn't really know a lot about vaccines, what do you have to say about all that?
So,
Sorry, as a listener who
Sure.
doesn't know a lot about vaccines, talking to someone who does, tell me about that.
Well, we've made a lot of virus vaccines successfully, and we've eradicated smallpox using vaccines. We've almost eradicated polio, and now there are, over 80 companies making different vaccines. One of those is going to work, probably more than one. So I'm pretty confident that we can have a vaccine in 2021, and I think it'll work. I really,
Oh, that's cool.
I don't see any reason why it can't work with so much. I mean, this is the thing. This country, the world, has so much scientific brain power. It can do pretty much anything if it puts its mind behind it. The problem is when you don't put your mind behind it and you languish and don't do anything. So I think a vaccine is going to happen. It'll be tested this summer and this fall. It'll be rolled out in 2021. And so that, at the most, we'll have one more round of outbreaks this fall. And then next year we'll have a vaccine. I don't think that's an issue at all.
And I know that to Monday morning quarterback, something is real easy, and in the moment, it's really hard to know what to do. But going back to the beginning of this outbreak, I just want to get your thoughts on how it could have been handled with hindsight, and also, the idea that this could have emerged from one of the Wuhan labs started off kind of a shadowy conspiracy theory thing, but it's been really pushed into the mainstream at this point. So I wanted to get your thoughts too on whether, because definitely there's emerged some stuff about NIH funding back coronavirus research in Wuhan, and that these labs might have had some poor biosecurity issues, and that SARS has escaped from a couple of Chinese labs in the past. Do you think there's any validity to the idea that this might have been something that walked out of a lab?
Oh, zero validity. And if you think it, you're just not looking at the information, or you don't. I mean, I think part of the problem is people don't understand much about viruses. So I don't know how they could believe that. So someone told me yesterday that 30% of Americans believe that this came from a Chinese lab. And I said to her, 30% of Americans, 99% of Americans don't really understand viruses. How can it be that they think they know enough to make a decision? Yeah. The data are very clear. This is a bat virus. It's a brand new virus. No one had this in their lab. Now, you could say, well, maybe the Chinese lab had it and they didn't tell us. Well, that's an accusation. It's not based on any evidence. The evidence we have the genome sequence of this virus, and it looks pretty much like a virus from bats harvested in 2013. And so probably this SARS-CoV-2 and that bat virus had a common ancestor, and it just infected a human somewhere outside of the city, perhaps, and started the outbreak. And the only way, I mean, if I sit down with you and I show you these genome sequences, you'll believe me in the end. The only way we're going to settle this is to do a lot of wildlife sampling and find the precursor of this virus. Uh-huh. And then we'll say, oh, you see, it's in bats or it's in some animal in the countryside. And we haven't done that very much because it's hard to get funding for that kind of field work. So I think these theories, and you see some prominent virologists saying, yeah, it could have. No, it really couldn't have. It did not come from a lab. It's quite clear to me, and I think these accusations are useless.
Well, not that it was built, not that it was like a recombinant virus, but that it was just a virus that had been taken from a bat reservoir and had been worked with or looked at.
Yeah, the building is not possible, right? Nobody knew how to build this. This has
Okay.
features that no one would have guessed. The escape from a lab, well, if they were working on something like this, exactly, it could, but nobody had this virus. And, scientists don't hide stuff. They publish stuff because that's how you get notoriety,
Well,
right?
I'm not trying to argue for this. I just want to. The Chinese aren't exactly not the people of China, but the Chinese Communist Party isn't exactly known for being forthright with the world community. So I think that's what leaves some people feeling a little bit. Because, like, what are
But
your thoughts on their what are your thoughts on their numbers that they're reporting?
well,
Because they seem just absurdly low.
remember that, but the Wuhan Virology Lab is its own thing. They get to publish what they want, and they don't have to report everything to the Chinese government. And I know they don't. So they have steadily published bat coronavirus isolates over the years, and there's no reason to expect that they would withhold one that just happened to start the
Right,
pandemic.
okay.
It doesn't make any sense to me whatsoever. Now,
Okay.
in terms of, so I don't think this is a virus. And in fact, if this came out of the Wuhan lab, why didn't they get sick? Why did people in the market a few miles away get sick first? That doesn't make any sense.
I thought they were saying now that patient 0 was an employee at the lab.
Now, that's just hearsay. It's not that person
Okay.
didn't even have it. They probably had influenza or something. No, there's no SARS-CoV-2 in that lab. That's the whole point.
Do you think China's numbers have been accurate as far as what they've reported for infections and fatalities?
Probably not, because I think it's impossible with so many people to be accurate in how many cases and fatalities you've had. I think they've done the best, but I wouldn't be surprised if it changed. But I'm not sure it's been done willfully. I think they were simply overwhelmed with the number of cases they had. And, the same thing happened in Italy. They got overwhelmed as well. Now, you asked me something that we didn't address. Maybe we can get back to it, but, what would we have done differently? So let's not forget that. You can ask your question, but let's
no, that's actually what I was just going to ask you, sort of going back to the origin story. What, what could have been done that is going to be obvious to us in retrospect?
So I think some countries have done things that should have given us the lead, and part of it is having a plan. So if you see a new virus starting to circulate, to the extent it was in China already in December and January, you put your plan into place. And that plan includes not just having hospital capacity and PPE, personal protective equipment, stockpiles and so forth that you can release. We had none of that. No preparation for ventilator requirements, but testing. You have a plan in place to have X number of companies. We have wonderful tech in this country. We could have had 50 ready to go and design a diagnostic test that could then be deployed at massive scale already in January. And remember, this is when the CDC starts fumbling with its own test that was bad for a month, and then they had to reissue it. And finally, in March, they say, okay, you can make your own test. What a blunder that was. We should have had 50, 100 companies in January. The federal government should have said, here's the virus, make a test. And then we could have distributed it to all 50 states. We could have said, there are foci of infection in Washington, San Francisco, Chicago, New York. Let's quarantine these people. We could have stopped it. We didn't. That's what I would have done. If I were the SARS czar, I would have done that. But, there's
You
no
think it was too
impetus.
decentralized, and there, are you saying, like, because one of the questions that I've wondered about is how there's been a lot of talk about being unprepared, but if it's a novel virus, then obviously you don't have testing in place. So you're saying that what you would do is create the test and then disseminate that out to all the companies that could have been standing by to produce those tests?
Well, the companies that would have the expertise to make a test, right? And then as soon as the genome of the virus is released in early January, say, here's the genome, make the test. We want X million tests in a week, and we're going to distribute them to this state. So you need top-down directory for that, right? And, our current administration doesn't believe that. They believe the states should be doing everything, but not in a pandemic. You can't have the states. They don't have the expertise to do that. Some do. New York did, but, Idaho doesn't have the expertise, and they're going to get left behind. So in some situations, you need top-down directives. You need a pandemic plan that's going to do all this, and we didn't have one.
Yeah, it's really interesting. So how do you feel about the social distancing guidelines, the way that we've shut down the stimulus bills, all that kind of stuff? Looking at all that, if you were the COVID czar, coronavirus czar, how would you have handled this? Knowing that this gets a little bit out of your area of expertise, of course, I'm sure.
I would have put the money into coronavirus and other virus research long before, and it would cost less to prevent
Yeah.
it than to recover.
Yeah.
But this is hindsight. So from the first, I would have put out a directive. We have to do social distancing in all states, not just the ones that want to. You need to stay home for the next month. Businesses have to shut down. And we're going to take this on a week by week basis and see how the cases go. It's been piecemeal, right? Some states doing really well, some states doing nothing. And that's where we are. But that's what I would have done. I would have certainly had a directive to say this is how we should be dealing with this. Now, this is unprecedented, of course. The other approach is not to do anything and just let the virus run its course, in which case many, many more people would die because they couldn't get hospital care. So you have to make that decision. I would decide in favor of saving people's lives and then at some point deciding when. It's never going to be 100% safe to go back to work. There's always going to be a threat. But as I said, you have to balance that versus wrecking your economy and your social structures.
Where do you weigh in on the public using masks, particularly, I guess, fabric masks and things like that as a means of reducing the amount of people that they kind of get their droplets on?
I absolutely support the use of masks. Now, early in the outbreak, I didn't because the CDC and WHO said not to wear them. And I believed them because I had faith in them. But it turns out they actually work. And only in the last few weeks, they have said, yeah, you should wear them. So I wish from day one people had been wearing them. I wear them whenever I go out. So I think, yeah, you should wear face masks when you go out.
Do you see that? I remember, I guess maybe 10 years ago, coming home from a flight and getting online and just Googling, like, why do so many Asian folks wear surgical masks in the airport? I didn't understand. Yeah. Now it makes more sense to me, how it's become a cultural thing there. Do you see that ever becoming part of our culture here? And I guess the bigger question is, do you think there comes a point where we just forget all of this, or do you think that this actually becomes part of our cultural identity moving forward?
It's hard to say. I've had conversations with people who want to make fashion face masks, things that look good that you can wear and are more. I mean, a face mask is very awkward, not easy to put on correctly. And you can imagine designs that would be much, much better and look good. So it could. It really depends how much this pandemic sticks in our consciousness. And if it just, it ends next month and that's it, we never hear of it again, then I think not. It won't be part of our culture. But and I think most of the people wearing masks in this country are in very specific areas like, the Northeast, the West Coast, and so forth. So I'm not sure it's penetrated everywhere yet. So if I were to guess, I would say no, unless something very different happens.
I kind of enjoy the sort of feeling of anonymity. It's
yeah.
I kind of wish it would stick a little. I'm going to give you a list of names or organizations, and I'd love if you could just give me your, A+, A, B, C, D, F, whatever. Give me a grade on how they've handled this, and then feel free to comment on any of them if you want to. The WHO.
WHO, I give an A-. I think they've handled it generally well, except for the face mask business. And I think they probably should have declared this a pandemic a bit earlier, just to help mobilize resources.
How about the US CDC and NIH?
So NIH, I would give an A+ because of Tony Fauci, because I think he's great. And he's handled this really well in the face of administrative opposition. He's a good clinician scientist, and he knows. And I think he really is interested in saving people's lives. The CDC, I would give a B, unfortunately, because although I know lots of good scientists there, they blew the face mask issue and they blew the testing issue. They should have distributed the testing to other institutions very early on in this outbreak.
How about the Presidential Corona Task Force?
I
know that's a complicated mix of people.
Yeah. Well, if you take out Tony Fauci, of course, gets an A+. The force gets a C. I don't feel that they've done anything to help anything. Maybe they should get a D, actually. They haven't done anything to help the situation in this country. I think what they have done is for show mainly, and they've never issued any directives. They've never helped any particular state with testing, and they've completely failed at controlling this outbreak. If it ends up killing fewer people than we thought, it's only going to be because of certain states taking the initiative.
President Trump.
Trump gets an F.
Now, are you somebody who would give him an F on everything anyway, or is this, like, because he's such a polarizing figure that there's a tendency for
Yeah.
people to hate and love, right? So, are you on the going to give him an F no matter what, or you feel you can say that genuinely based on just his response to this?
Well, I mean, I don't hate anyone. Okay, I don't hate him, even though I think he's not good for this
Right.
country. I don't hate him, but I think he's overall done poorly. He doesn't really have the people of this country in his best interest. But if we just focus on the outbreak, I mean, it was just a month ago. He said it would be over soon. He completely didn't listen to Tony Fauci. And I don't think he's done anything to mobilize help. He hasn't gotten testing mobilized. He said, "Oh, I don't think there's any problem with testing." So I think we needed a real leader here. And his vice president is marginally better because he knows how to organize people. But I think Tony Fauci should have been the guy heading the task force, and he should have been allowed to do what he wanted to. And the president should have stepped back. So independent of what I think, we could, I would have to go over everything he's done. I probably wouldn't give him an F in everything, but in this public health crisis, he gets an F.
And I've actually heard you say, I've got some more names here, so I'll move through this quickly. But I've heard you say that because he's obviously really played up the shutting down travel to China. But I saw you say something about feeling like that was a little too late anyway.
Oh yeah, that was not very long ago that he wanted to shut down travel, and it was too late. I mean, the key was to get testing done here to see what's already here. And we had all these foci of infections we didn't know about, and that's what got us in trouble. And if we had picked those up earlier, we could have solved it. But the travel restriction was just too little, too late.
How about Deborah Birx?
I listen to her, and I hear a lot of conflicting things. The other day, she said, "Well, there's a lot of places that haven't had any infections, so let's open them up," which makes zero sense to me. Yeah, if they haven't had any infections, then they're all susceptible. So why would you open it up?
Right.
You could open everything up if you wanted to, or maybe do some testing and make sure that that's true. So I'm not a big fan. I would give her probably a C.
How about the U.S. press, U.S. media?
The press gets probably a C minus, which excludes you, of course.
I don't consider myself part of it.
I think they just sensationalize this. They don't provide people with any information. And I have to say, we get lots of emails on Twiv, and most of them say, we turn to you because we can't get the scientific truth anywhere else, including the mass media. And so I take that as a failing on the part of the mass media. It's all sensationalism. How can you have 24/7 coverage of this thing? It's crazy, and nothing else.
Right. Almost nothing else.
Almost
right?
nothing else.
Yeah.
And just a stream of people saying different things and no cohesiveness. I think that, well, the media, as you said, is a for-profit proposition, and that in itself is a conflict, right?
How about the American public in general?
Oh, well, I would give them a B because not enough listen to Twiv or listen to my lectures. There's no excuse not to learn more about viruses and be able to understand what's going on here. I think I know it's hard to learn tough subjects. It takes time, but everyone's capable of doing it. And I think people should have sought out information a lot earlier, and not just in January, but in previous years. You should understand the world around you. You should understand, like you like to talk about your ecology, and you should understand all the microbes that are in the world and how they might impinge on you. And so for that, I think they get what did I say? A C?
You said a B.
A B. Yeah, I'm being generous because, a lot of them are dying here, and I don't want to blame them. But I do think the bottom line is, please, the internet is an amazing thing. You can learn almost anything, and there's, free courses and free podcasts. Take advantage of them to understand what's going around you.
I'm going to say, I'm taking university classes from you for free. So it's incredible. Last question, and then we'll go through sort of where people can find more of your work. But what viruses loom on the horizon that have your attention? And, I feel like so much time had gone by since we'd seen a real pandemic that we got lazy and we got tired and we didn't maintain the stock, and all those things happened. We got complacent. This is going to wake us up for a little while. And while we're awake to it, what viruses should people be looking at, or what classes of viruses?
Well, unfortunately, the ones are the ones we know about because we don't do much
Right.
field and wildlife
Mhm.
sampling. And so we know bats have a lot of viruses in them, right? Bats are 20% of mammals, the most numerous mammals. They have more viruses than anything else on the planet. And so they're an obvious place. We know lots of SARS-like viruses in them. There are viruses of different families like Nipah and Hendra, which have come from bats in Malaysia and Australia. So there are bats in Africa that are problematic. They seem to harbor the Ebola viruses. They have the viruses that went to camels to start the MERS outbreak. So I think we need a better sampling of those viruses in bats. There are also a lot of viruses in rodents, and most of which we don't know. Hantaviruses cause a really serious respiratory disease, and that's a spillover from mice to humans. And that could take off and become a human virus as well. And we don't know what else is out there. So I think we have to look. We have to do more wildlife sampling, particularly in mammals that are numerous and that live near us, to find out what actually the threat is. But there are plenty of precedents for doing that because these viruses that I've just rolled off are just a few of the ones that come from mammals to humans.
I just want to say that I really appreciate your time today and you taking the time to go through some of these questions, which are a little sticky. And I just,
No
so
problem.
thank you. And tell people where, and we'll put links to everything that you say here, but where do people go to find your podcasts, your YouTube stuff? Where do you like to send people?
So you can, This Week in Virology, you can get on any podcast player, but if you go to microbe.tv, you'll find TWIV and all the other podcasts we do there. Those are where the show notes reside, and you can also subscribe there by email or RSS or whatever your favorite podcast player is. And then over on YouTube, that's where I have videos. I have my course, my virology course, and also when I used to travel, I used to do podcasts on the road at universities and meetings with guests, and I did videos of those. So those are all there. I have a whole bunch in the can that are ready to come out when people are not obsessed with coronaviruses anymore.
Right, right.
So the
Bad
YouTube
time to put out anything else.
might use, you can search for me, Vincent Racaniello or Prof VRR, PROF VRR. And so microbe.tv and YouTube are the main places. Of course, I'm on Twitter, Facebook, and Instagram also. You can find me where I mainly talk about viruses.
Last thing I want to ask you is, are you optimistic or are you pessimistic about the future of the world? Because this coronavirus is just one little piece of all the craziness going on in the world. So are you somebody who looks out at the future feeling like doomy and gloomy? Or are you somebody who looks out going, I think we can solve these problems?
Oh, I think we can solve them. We have amazing science capability, not just in the U.S., but the whole world. And I think we put our heads together. We can solve any forthcoming virus. We can be ready. It's just a matter of our will. So the tech is there. It's just a matter of whether people want to pay for it. And I think after this one, we're going to have some changes in how drugs are made. So I'm more optimistic.
Thanks so much for your time today, Vincent.
It's been my pleasure. I enjoyed speaking with you.
Thanks for listening to the WildFed Podcast. You can help us grow the show by subscribing and leaving us a rating and review. It ensures better rankings and more advertiser interest, which translates directly into better shows, more awesome guests, and a constant stream of fresh, new content. Have a question you'd like answered on the show, or a hunting, fishing, or foraging trip you'd like to host us on? Email us at info@wild-fed.com. And be sure to visit our website, wild-fed.com, to check out the WildFed video show and store. WildFed. Food is all around you.